Thank you, Emmanuel. What a wonderful delivery. And this is really a success story. And thank you for sharing that with us. So I would like to invite you and all other speakers to come to the podium. So we'll take questions because they're all done very well. We have 20 minutes questions. Please raise your hands, be brief, tell your name and institution. And if you have more details, we can discuss at tea time. So be brief, and also I expect brief answers from the speakers. So who wants to ask the first question? In the back there, yeah. Come forward, please.
Hello, good afternoon. My name is Dr. Bindu Madhav from World Vegetable Center, Ikrisat Campus, Hyderabad. My question to Professor Shashidhar, I like his talk on the root in rice, but I need to know what is the contribution of root. If you take for the total biomass, maybe 50%, but what is the contribution of root to the yield in rice under different conditions? Thank you.
Contribution of roots is first to maintain itself and maintain the shoot healthy and in a handsome manner so that it can produce grains when it is required. If roots were to be correlated directly with yield, you won't get a direct correlation because presence of roots by itself doesn't mean anything. How it functions, that's why I told you intelligent roots, effective and efficient. That is what matters at the final end. It's like correlating, if I'm permitted, it's like trying to correlate the presence of legs in a football player to the goals that is scored. You cannot correlate. Without legs, he will not perform. But at the right moment, at the right time, he has to use the legs in a particular way, then the goal is scored. So direct correlations will not exist and not present in any paper so far published.
Thank you. Mark Cooper.
Mark Cooper. Vijaya, I really enjoyed your talk. Thanks very much. My question is actually for Nayaba Temi. He's not paying attention, but now he is. I really enjoyed your use of the photoperiod sensitivity, really targeted it. I thought that was a really nice example of use of clear understanding of some of the traits and how you can target those to have a big impact. I was also interested in whether you could tell us a little bit more about the stay green genes, the QTL that you were looking at, and whether they may be associated with the root angle components that Vijaya was talking about, or perhaps there are others that are targeted towards moving water from vegetative development and making that more available for the grain filling period, because it seems like there's a lot of water resource that you're avoiding by getting the flowering time right. Maybe that water could actually be used to help during the grain filling period. Interested in knowing how you're targeting that.
You got the question? Yeah, you can. There's the.
Thank you for your question. When we built the sorghum population, we didn't look for QTLs, but the two parents we had some characteristic. From the first parent, Chandugu, during our phenotyping, we observed that it stays green until physiological maturity or beyond. But which kind of QTL is in that plant, we don't know. But we took care of that QTL in terms of leaf staying green after physiological maturity, because in our breeding process and in phenotyping, we are counting the number of leaves that stays green after the physiological maturity, and we score them and include in the recurrent selection. But which kind of QTL is there, I can't say.
Okay, thank you. Now, Dr. Bansal, then followed by this one there. Yeah.
My question is to Dr. Shashidhar. You laid emphasis on deeper root system, obviously, for better uptake of water, particularly under drought conditions, and you rated it with higher yield under drought conditions. But as a physiologist, I wonder that what are those factors contributing to this higher yield? For example, is it higher biomass? as you mentioned in your beginning as well, that is an important factor. Or roots not only take up water, also responsible for uptake of nutrients. So you think, is there a possibility of higher nutrient use efficiency as a result of deeper root system, particularly nitrogen use efficiency, certainly not many others, because they prefer shallow root system, while we talk only of the deeper root system, particularly with drought? Or could be both? Or you think harvest index also has a role to play when you talk of higher yield, particularly in a drought? So what are those contributing factors, really speaking, at a whole plant level? I'm talking about it, whereas there could be many others, and that would be useful if we can really analyze those. Because as you see down the stream, we are working on various aspects going down to the QTLs and also to the genes and promoters and those combinations. So it will be useful if you think you have developed some wonderful material that we can also try to identify those factors, at least in your experiment which you have done so far. Thank you.
All of what you said is right in different proportions, as indicated by high nutritive content of the less watered plants or aerobic condition are more nutritious than the well-watered conditions plants, same genotype. So there's something interaction that's happening at different levels.
Excuse me, sorry, I'm not talking of nutrition from the nutritional point of view. I'm
Oh,
talking
not
of
grain.
uptake of nutrients as such, nitrogen, for example.
Okay.
I'm not talking of higher zinc or another, those phytonutrients. No, I'm talking of nutrition for the plant.
So under aerobic condition, the nutritive utility of nitrogen specifically was lesser than the aerobic wetland condition because the runoff was not there. 70% of nitrogen in irrigated conditions is runoff, and it causes eutrophication of lakes. So under aerobic condition, not a drop goes away. So lesser nitrogen can be applied and more NUE is there.
Thank you. We got two questions lined up there. First, you, yeah. Tell the name.
Felix from Africa Rice, Nigeria. My question goes to Dr. Low. During your presentation, you gave a hybrid rice that gave maybe close to 13 tons per hectare. But when I checked the fertilizer usage, I said about 780 tons, kg. So I want to ask, is it tested only in China or Asimbi? Because when I check, the fertilizer used for me is very high. I want to ask, is it possible if you reduce the fertilizer usage, are you going to get similar yield? Secondly, for the variety developed through Ira-209, we know is drought tolerance. And we've identified it as to be drought tolerant in Africa. I want to ask the QTL that was developed, and it's like you said, maybe a gene was developed. On what was it expressed? Is it on roots, on leaf? I didn't get that very well during your presentation. Thank you.
Well, I think it's pretty hard to hear from, but the first question is that can you get high yield with low fertilizer use? Is that a question?
Yes, with
Yeah.
the hybrid.
The second question I lost, and third one I lost.
No, I only asked two. The second one is the gene that that he said it's like maybe they found a gene from the Ira-209 or a QTL. I want to ask, was it expressed on the roots or leaf? Or I want to know
Where
the
was
expression.
the gene expression, whether it is in the roots or the shoot, for the better drought tolerance? I'm putting in a crude way. That's what I asked. Okay, let's finish these two questions. Okay?
Yeah, I cannot kill canary. Actually, the WE and NUE is different. So in plant breedings, we are focused on the WE, the water use efficiency. Collection use efficiency is just collect with the cultivation. When we put less water in the field, so we can put less nitrogen, so not a genetic improvement. But we are trying to increase the NUE by identify some rice germ presence with the high NUE. The first question, right?
Yeah.
Okay.
So basically saying there has been nitrogen use efficiency by shifting into dryland production system. So less nitrogen fertilizer was used to produce high yield. Otherwise, a lot of nitrogen was lost in the system because of the flooded conditions and so on.
Yeah,
Yep.
it's completely different. And the second one is you just says the QTL the function of the QTL is different. One is for shoot and the other one is for loading for root development. The point I'm going I already said is that the drought avoidance is mainly come from the roots. So I still not find some QTLs can function both in the shoot and the root.
Thank you. Next question.
Rajinder Ranu, Colorado State, USA. My question is to the gentleman from Pulse Institute, Kanpur. I lost the name. I couldn't figure out what was the real name of the person. You showed a picture showing chickpea variety which had 6 seeds to a pod. If you cross that variety with the elite variety, what is the outcome? Can you share that if you know the answer?
Yeah, we have started some breeding experiments with that using Cicer cutinium with Cicer eritinum. And we have got some good segregants, which has around 4, not all 6, maybe 3 to 4 it's coming. We still need to see. And these are, of course, in the initial stages.
Are the seeds much smaller or larger, or what's the?
Yeah, seeds are smaller, a bit smaller.
So the answer is that the program is still early stages. Significant back crossing is required to get the seed quality, seed traits, etc., into the size eritinum. So next, please.
Graham Hammer, University of Queensland. I just wanted to ask Dr. Del Porro from Chile about the water-soluble carbohydrate. Sorry, the questions to Dr. Del Porro from Chile about the water-soluble carbohydrate work. I'm just curious, in Australia, there's been a lot of interest in that trait, and a large part of it seemed to be consequential on differences in tillering, and I just wondered whether you had looked at that.
Yeah, there is difference in tillering. Yeah, at the end, we found difference in a number of spike per square meter. So that's the reason, because there was a strong negative correlation with water soluble with carbohydrate. That is the reason why we don't found a positive correlation with grain yield. But there is a positive correlation with the other 2 yield components, which are number of grains per spike and kernel weight.
Well, I could just ask a supplementary question there. Did you measure the water-soluble stem carbohydrates in the main stem or the tillers?
Yeah.
And what's the contribution of the tillers, if at all they're in relation to mobilizing some of them stored in the stem of the tillers?
Yeah, we measure only in the main stem, not in the tillers.
One question from the back, please.
Lucky Omoigwe is the name from IITA. My question goes to Dr. Pozo, who works on the water soluble on wheat. I want to know if the expression of the gene was at the early stage or the later stage of growth of the plant, because we also observed a cowpea for swollen stem that has a very high water soluble carbohydrate. But when you compare it with the susceptible material, there wasn't any difference, but we could only find a difference at the later stage of the plant growth. Did you also observe this similar, did you have a similar observation with your own study?
You repeat that question again. We're finding it very hard. Very briefly.
Okay, I mean the stem, the water soluble carbohydrate in the stem. I've been in cowpea. We found that for the swollen stem, the expression was not seen at the early stage of growth, except at the later stage. But I want to find out if the same observation was observed in wheat.
I think I partly heard the work you have done in cowpea. Is that right?
Yes.
Yeah. So early stages.
We didn't find any differences, yes.
Any differences, but
Yeah.
later stages you found.
Right.
So the carbohydrate accumulation in the stem.
Yeah.
Yeah. So he's asking the question whether, well, what he showed was that the maximum was around 10, 12 days after anthesis. So in the case of wheat, the carbohydrate accumulation moved slowly and reached the peak just immediately after anthesis, being a determinate plant. So that's the answer, correct? Francois.
Yeah, my question is to Vijaya Singh. There is a very strong genetic control of the angles. That's great. Is it completely constitutive, or is there...
Francois, can you come a bit more closer to the microphone and speak up, please?
Sorry, I do that again. So there is a very strong genetic control of the angle, but is this completely constitutive, or is there some contribution of adaptive things depending on the environment? And second thing, could you elaborate a little bit where you would like, in which scenario you would like a wide root system, and each scenario you would like to have a narrow one? Is this clear?
So actually, because of the time constraint, I didn't present most of my other data, but what we sort of simulation studies have shown that the narrow angle lines will be more sort of for solid planting, whereas the wide angle lines are more common for skip row planting, which is more common in Australian environments. So this how we can use the management system to increase yield under different environments. And sorry, I didn't get the first question. This was the second question.
Is it completely constitutive? So one genotype has one angle and that's it? Or is there some degree of adaptation to environmental conditions?
Doesn't look like, because of the high heritability, we haven't seen, because we've done this in different size of chamber and it's quite like it's maintained over and different soil, it's same. So it looks like there's not much plasticity for the root angle.
Okay, Victor Sadras.
Yeah, I would like to comment on that, because true that angles are under genetic control and it's strong, but the trait is density dependent. So if a plant is isolated, it would have roots like this, and if there is a neighbor, it would respond to the neighbor. So if you measure that in different plant arrangement, the angles will change.
Okay, there's one coming there.
I have two comment and question. One is to Dr. Singh and Sashida. Sorghum and rice is quite different in the potential root length. Both studied on root. In case of sorghum, how important to prioritize the modification of root? That is the first question and comment. And the second is...
Maybe let Vijay answer that question.
Oh, yeah.
So if we are sort of looking for drought tolerant, looks like with what I got, this nodal root angle and the vertical root, like narrow angle, is sort of more important for drought situation. Looks for this particular study, that's what I'm saying, and this particular trait. So that's the question.
Yeah, so Shashidhar mentioned that the root is quite important. In case of sorghum, root is as important as in case of rice. That is my question.
Yes, it is. and it's more extensive. You can. Did you see the root museum that we had in the poster session? We have museum of roots of all crops, all edible crops that we have, in fact castor also. So every plant invests on the root and the shoot in different proportions, and every plant thinks differently. It is probably related to its evolutionary history and genome size and many other things. But every plant knows how much to put to roots, how much shoots, and when.
Yeah, I know importance, but to what extent important might be different depend on the species. That is my comment. But let me ask a second question to, we hear the success story from China and India. I am very impressed. But I feel in Chinese case, using the hybrid rice, which has a bit higher yield potential, so the target yield environment in case of Chinese study and Indian or other cases, I think a bit different. About this aspect, would you like to comment?
The percentage of irrigation in China is, I think it's 33%. 33% of land is irrigated, assured irrigation. I may be wrong. You may comment on that. Yeah, 33%. So in India, it is far, far less. Depending on which state you come from, the irrigation is not so well. Infrastructure of irrigation is not so well developed. So it is more stressful here. Our environments are more unpredictable, and hundreds of years of cropping has depleted the soil, and Liebig's law of minimum has set in. Nutrient level of soils is very poor, so our figures are less. Yield figures are far less because of this.
Thank you. My question goes to Pozo, if I'm right to say. The speaker who talked about variability in stem carbohydrate mobilization. Well, before that, I want to say one word to my advisor who is here, if I'm allowed. Yeah. I have my advisor here by the name of Henry Nguyen. He paved my road to where I am, and I wanted to say him thank you, and I congratulate you for your effort for me being at that place. And I have his colleague, late colleague, Dr. Daryl Rosnov, who helped me work on sorghum. He's now gone, and I want him to say thank you. My question is regarding Pozo. I had problem screening a lot of lines in for water limit or water food irrigated condition. He's been screening 384 lines, and I want to know what kind of irrigation design, what was the maturity level of those lines under the same irrigation condition. Is there is any differences in anthesis, how carbohydrate assimilation happens? Is there any difference linked between developmental growth or stage of growth and carbohydrate accumulation?
We choose germoplasms with small differences in phenology. There were some few lines with late flowering we didn't include in the analysis, but because there is an effect. When you take the samples, the growing stage is important in terms of carbohydrates in the stems. For the irrigation system, we use the facilities of the INEA research station in Chile. They use furrow irrigation, and soils are a very good quality, and that's why we got such high yield in under irrigated conditions. But it's furrow irrigation.
Okay, we can discuss that later. So I think there are no more questions. We are on time. So I would like to thank the speakers. Please join me in thanking the speakers. And also you for attending and raising a number of questions. I think we have seen again some success stories. These stories needs to be perpetuated and told other places, because always the success stories are good to be told what went behind it, and the breeding for broader adaptation and drought has been successful if you look at the last 10, 20 years in different parts of the world.